Our next presentation is from Meghan Howard. Meghan is a PhD candidate in ecology and evolutionary biology, and she's going to take us back to Costa Rica and talk about the impacts of land use on Aedes albopictus distribution in Costa Rica. Meghan.
Great. Thanks everyone for being here today. I'm a second-year PhD student in the Mordecai lab here at Stanford, and today I'm going to talk about some of our recent work on land use and Aedes mosquito distribution in Costa Rica. So the Earth's surface is currently undergoing a massive transformation due to human-driven land use change. This map shows deforestation from 2000 to 2016. And although it captures a variety of types of land use change, it really drastically and powerfully demonstrates the scale at which we're altering the Earth's surface. And recent studies have started to connect this global land use change to changes in vector-borne disease. And a major priority has been to look at changes in the distribution and abundance of insect vectors because of the important role that they play in transmission. Just to give a few examples, land use change has recently been linked to changes in the distribution of mosquito vectors of malaria and tick vectors of Lyme disease. And so a unifying theme of these studies is that the response of different vectors to land use change really depends on the unique ecology of each species. Does that change in land use shift microclimate and habitat conditions toward or away from the range of suitability for that species? In our lab, we've become interested in studying the response to land use change of two species that cause devastating morbidity and mortality worldwide. These mosquitoes transmit diseases like dengue, chikungunya, and Zika, and they have some important ecological differences. Aedes aegypti is very closely associated with humans and is a highly urban species. It's considered to be the primary vector of the diseases that I mentioned, and it's also been very well studied. Aedes albopictus, on the other hand, is a more rural species that it's tolerant of slightly cooler temperatures. And although it's been demonstrated to be competent as a vector of these same diseases in laboratory experiments, its role in the field and in natural epidemics is less well understood. It's also the most invasive mosquito species in the world. And so there's much to be learned about it in its new ranges, including in Central America, where it's been invading in recent decades. And so we took an amazing opportunity, thanks to Gretchen Daly, to study these two mosquito species in Costa Rica. And Costa Rica is a really fitting place to do this work as a dengue endemic country that annually experiences a fairly high burden of dengue cases. This past year, the incidence rate at the national level was 110 cases per 100,000, although it was as high as 1,400 in a single county. And Costa Rica is also an interesting place to study land use. As you can see in the inset from that map I showed you earlier here, Costa Rica did experience quite a bit of land cover change in this period of 2000 to 2016. But, as Gretchen mentioned earlier, there's some interesting nuances to land cover change in Costa Rica. As of 1996, mature forests have been protected by federal law. And a lot of the crop expansion since then has actually replaced pasture or tree plantations or secondary forest. But even these types of land use changes might impact mosquito-borne disease. And as a global leader in sustainability, this is a really great country for us to connect this type of work directly to policy. They've been leaders in pioneering new frameworks for environmental protection, including paying landowners. This is the program Gretchen mentioned earlier. They pay landowners for the services, ecosystem services provided by uncleared forest on their lands. And these services might include greenhouse gases, mitigation of greenhouse gases, regulation of water quality, protection of biodiversity, and a lesser known but potentially important service might be protection of human health. And so we're currently pursuing this research question in Costa Rica. How does land use impact Aedes vector distributions? And we hypothesize that land use impacts on microclimate, vegetation, and breeding habitat will ultimately shape Aedes vector distributions in this landscape. And we decided to pursue this question with a very quantitative approach by using species distribution models. And these models incorporate publicly available mosquito presence data and remote sensing environmental data that describe surface features like temperature and land cover. And my collaborator, graduate student Chris Anderson, shown here, plays a huge role in this piece. And so using these maps derived from the same data we use in our models here, we can start to see these relationships between surface temperature on the left and land cover on the right. These insets at the bottom here show two cities in Costa Rica and really illustrate this relationship pretty starkly. We can start to see that generally in these urban zones, you experience much higher surface temperatures relative to surrounding vegetated areas. And using this approach with these species distribution models, we can generate spatially explicit predictions for occurrence. So this map shows our predictions for occurrence for Aedes albopictus across Costa Rica. And there's some important limitations to using this approach alone that I want to mention. There might be sampling bias in the mosquito data that we're using. Furthermore, this is presence-only data. We don't actually have true absences that we can incorporate. Furthermore, we can't really distinguish land use types with the type of data we're using right now, and that might play a role for these vectors. And finally, we have a limited ability to assess breeding habitat, which we know is really important for these vectors. And so, to address some of these limitations, we designed a complementary field study to actually go out into Costa Rica and trap these mosquitoes in the field. And this is a picture we took with a drone to really show you that our study region is this complex matrix of forest fragments, plantations, you can see this coffee on the bottom, pasture, residential areas. And so I also want to highlight another important plantation type, and I've included in this video for scale. This is oil palm. And these plantations can go on, as you can see here, for miles and miles. And globally, oil palm has been implicated in a great deal of deforestation in mature forest, although this likely replaced a banana plantation that failed in the mid-20th century. And our models can't really capture land use types like this and others that I've shown you. And so we went out into the field and extensively trapped in 39 sites across a gradient of land use and also a range of predicted suitability from our models. And this effort included a lot of help from undergraduate student Jameson Omar and graduate student from the Daily Lab, Jeff Smith. And so we combined 5 trapping methods and measured microclimate in each of these sites. And a lot of this work was supported by a Hellman Fellowship from Stanford. And so I want to highlight some of our results assessing microclimate differences across these land use types. So although minimum temperature at night was fairly comparable across these sites, we really started to see differences in the daytime with maximum temperature. And so ultimately, agricultural sites and residential sites had a wider range of temperatures that they experienced relative to forest sites. And as we expected, forest sites were generally cooler overall. And these temperature differences are really important when we talk about mosquitoes because like other insects, they are highly constrained by temperature. As shown here in laboratory data on Aedes aegypti, some key performance traits for mosquitoes are dependent on temperature. And the relationship is nonlinear. Values decrease at low temperatures, and again, often sharply at high temperatures. And so most of these species have thermal performance optimums for all of these traits. And so another thing I want to point out is that mosquito transmission of pathogens is also dependent on these same traits. And so the transmission process overall is temperature dependent. And so we expect that while Aedes presence and survival is dependent on temperature, we also expect that where they occur, their ability to transmit pathogens is going to be temperature dependent. And so we did find Aedes vectors as we expected in our field sites. Although interestingly, we only found a single individual of Aedes aegypti in exactly the type of site that we would expect. This site, shown here, was our hottest, most urban site. But then Aedes albopictus, on the other hand, was widespread across the landscape. I show here the average count of Aedes found on each land use type. And our resultant oil palm is particularly striking with these really high relative numbers of Aedes albopictus individuals. But our resultant forest is also really striking. We found absolutely no Aedes in any forest or forest edge site. And we also found 2 new important breeding habitats in these agricultural sites. So up on the top here, bananas are grown in both residential and agricultural sites. And they drop their leaves, and these leaves collect water. And in multiple sites, we found larvae in leaves exactly like this. And we also found larvae in agricultural tarps like this in the bottom, where they collect water as well. And so overall, we found Aedes albopictus in generally warmer sites. On the x-axis here, we have mean temperature in our sites, and on the y-axis, Aedes presence and absence, and this curve is just a simple logistic regression. And in red, I've emphasized these low temperature sites to show that we really have mostly absence at these low temperatures. Not to mention, these are mostly forest sites down here, as I mentioned earlier. These are the cooler forest sites where we're seeing a lot of absence. And then we also see Aedes albopictus in a wide range of vegetation cover, except for at high vegetation cover. So here I want to emphasize that regardless of land use type, at about 80% cover or higher, we see absolutely no Aedes vectors. And so we think these results have important implications for transmission. We see dengue cases in this area where we actually found no Aedes aegypti, and so we think this suggests that Aedes albopictus could have an unrecognized role in local disease transmission. And we also think this suggests that forests may protect against dengue by limiting these vector populations. And we don't fully understand the mechanisms behind this result yet. We expect that temperature and habitat might be prohibitive of these vector species in forests, but there could be other factors at play, including abundance of predators or competitors, or access to hosts. And so we see several next steps ahead to pursue. First, what is the role of Aedes albopictus in transmission of dengue in Costa Rica? And we're currently testing our samples for virus as one way to pursue this question, but we'd also like to look into case data. Secondly, what are the mechanisms underlying the protective effects of forest? And one way we'd like to pursue this question is by using available biodiversity data from these sites to try to assess differences in predators and competitor abundances. And finally, how do climate and land use interact to shape disease transmission? And our team is ultimately going to develop a new disease risk modeling approach to try to answer this question. And so we're excited to continue generating collaborations with Costa Rican researchers and policymakers and groups like the NatCap initiative to try to connect this work to a larger framework in thinking about ecosystem services for health and land use planning and policy development. So I want to thank Gretchen Daly's lab once again for the opportunity to pursue this work. We've really enjoyed this and are really excited about it. And also to your team for all of their support. And thank you all for your attention. I'm happy to take questions at this time.
Bob, please. I should have asked earlier, questioners, please identify yourself.
Bob Siegel, microbiology. Cool presentation, cool videos. Given the fact that Aedes albopictus is an introduced species, and given the fact that people are now actually seriously talking about using gene drive to drive local populations extinct, can you comment on what ecological services Aedes albopictus provides in these areas?
Yeah, so ecological services and beneficial services? Yeah. Interesting. Yeah, this is a point of ongoing debate as to what are the benefits of having mosquitoes. I guess there's several parts of your question that I would like to speak to. Driving Aedes populations down generally is going to be very difficult with the new technologies we're developing in the near future. And so I think I want to emphasize, first of all, that it's really important to think still about using vector control and understanding the ecology of these species. This is something that in most places of the world, it's going to be a while before we can use these kind of new developing technologies to alter vector populations. And so it's really important for work like this to first understand where do we invest our resources in vector control. And then the second half, in thinking about benefits of having mosquito populations, I think that's a great question, and I frankly am not aware of much. I mean, they're a food source for some birds and bats, but generally don't seem to be a large proportion of the foods that they eat based on diet analysis. And so I think I'm on the side of really thinking it's okay to, if we can, drive these populations down as much as possible.
So a question for you. Mm-hmm. Where is the sylvatic dengue transmission in Costa Rica? I mean, dengue is known to emerge from sylvatic cycles.
Right.
And if there are no Aedes albopictus in the forest, are there no non-human primates there? Has that whole cycle gone away?
Yeah, so there are some in this region, but actually very few. There used to be more, and there are thoughts that maybe through a great deal of land use change in the past century, really, most of the non-human primate populations are elsewhere now. However, in another region of Costa Rica, there are quite particularly in a very high dengue burden area, there are a lot of non-human primates. And so there's probably likely there are many other mosquito species out in the forest. And it's very likely that, like for yellow fever, where there are important bridge vectors that transmit between primates and people, there are likely other vectors playing that role. And I can't really speak to this area. However, I will say that our trapping effort involved sampling beyond just Aedes species. And so we're actually going to be looking into this to see what other species were in our traps and what do we know about their role as vectors, or what don't we know, for that matter. Okay.
Gretchen talked earlier about working to engage policymakers around questions of ecosystem services. And you rather provocatively suggest, and the data suggest, that land use impacts these disease vectors. I'm interested in your perspective on the local opinions and attitudes towards the research you're doing and the results you've generated to date.
Yeah, it's a great question. So all of the work we do down there, obviously we're trapping across almost 40 sites, which means we're working directly with landowners, and they're giving us permission to come onto their land or their plantations to do this work. And so we did have a lot of conversations about this. And generally people, particularly of note, those who are growing crops, are very interested in the results of this work, or interested in trying to think about how they can tailor management practices or protect certain features of their land, like forest fragments, to try to promote these ecosystem services. And so one thing I can say about Costa Rica is people are very interested at every tier, all the way up from the national government all the way down to local landowners, in thinking about these things, which is really exciting and hopeful. And so, yeah, I think it's the kind of place where this is really novel territory, but also a great kind of first trial at these kinds of things, and incorporating that into policy and management practice.
Question.
Hi, I'm Felix. I have a question about absence data. So as mosquito populations are super heterogeneous and ephemeral, how much data do you actually need to conclude that something is absent? And do you think that's hard in drawing any conclusions?
Yeah.
This type of work?
Absolutely. So I mentioned these publicly available data, which are obviously only presence, and so we have a ton of that. But this is, what we're going to be using for our absence data to try to test this. And so I think one thing we can't be really sure about here is that there's a lot of, particularly with Aedes albopictus as an invader, a lot of uncertainty around kind of, is an absence really an absence because of suitability, or is it because it hasn't quite reached this suitable territory yet? There's a mix of things happening in this study site in particular. And so I think one thing that might be worth pursuing is having multiple regions sampled. And we might do this as well, but actually try to capture in slightly different ecosystems or regions of countries like this to try to assess where those absences are versus presences, and maybe relate that to where they've been invading in recent years. So the simple answer to your question is I think it requires a lot of field sampling, which is a very hard thing to do. But yeah, it's a good point, and I think this is something we're working on now, is what really counts as a pseudo-absence versus a real absence in these models. If you have thoughts about that, of course, be happy to talk.
Thanks, Megan.
Yep, thank you.